Welcome to Plutus IAS, and today we are covering sodium-potassium pump. So as we know, the sodium-potassium pump is an important topic, one of the most important one, especially when it comes to your bioinorganic chemistry. So for the bioinorganic chemistry, it is related to your paper one. The sodium potential pump plays a crucial role in the traveling of nerve impulses throughout our body. In this, in our cells, there are two portions. One which is the inside part of the cell, which contains the cytoplasm, and the outer portion of the cell. In the outer portion of the cell, the concentration of potassium ions is much larger compared to the concentration of sodium ion is much larger compared to the concentration of potassium which is are inside the cell. So what happens is, in a sodium potassium cell, because of the difference in concentration across the cell membrane, there is a concentration gradient that is being produced. Since both sodium and potassium are your alkali metal, they exist in charged states, the difference in the concentration results in different ionic activities, which also results in generation of charges across the cell membrane. So because of which a current is being produced, which is being produced due to the concentration gradient. And because of that, we see that traveling of nerve impulses from one section of the body to the another. In this process, we will be utilizing an ATP for the energy purposes, in which magnesium will be playing as the active site for the process. Whenever we talk about sodium potassium cell, this is a general idea. This is a general outing of the cell where this portion is your cell membrane, and this is your sodium potassium ATPase molecule. This is a tetramer unit. This is a tetramer unit containing two alpha portions and two beta portions. Now, in a normal scenario, at rest position, the sodium potassium, this molecule is in a closed conformation, which is known as its even configuration. In this even configuration, the phosphate ion along with an ATP molecule is bound to it. Now, since inside the concentration, inside the sodium ions have higher concentration, while outside they are in lower concentration compared to potassium. So initially, the sodium ions has inside of the cell, there are active sites which are specifically reserved for your binding of three sodium ions. So initially, three sodium ions bind to it, and there will be a change in polarization across by the binding of Na plus ion when they bound to this alpha and beta units. Now, later on, this alpha unit, even after the binding of Na plus ion, is still in a closed conformation. So what happens is, later in the process, this phosphate ion from the ATP, it will undergo, first we'll have phosphorylation. Phosphorylation. In phosphorylation, the ATP will break down, sorry, ATP converted to will get converted to ADP, which is adenine diphosphate, along with the inorganic phosphate group, and releases energy. Now, because of release of energy and change in the polarity due to the binding of Na plus ion, now this molecule turns open, which means now here these two portions will come closer and the outer portion will start to open. Now, what happens is, as it goes to the outside, at this process, the Na plus which are bound to the outside are now exposed to the outer portion of the cell. In the outer portion of the cell, now the other portion which had sites for binding of the potassium can come across, where now because of the energy, it's not just the shape of the molecule that has been changed, it also changes the polarity, because of which 3 Na plus ions will be lost to the outside. When 3 Na plus ions are being lost outside, it is called as your E2 conformation. It is called as E2 conformation. And then 2K plus will bind to the inner portion of the molecule, and then again the polarity changes because the binding of K plus will be different from your Na plus. And since also the number of ions that are binding are also different, so because of which the polarity will again change. But still, the E2 conformation at this point will still remain as it is because it requires a higher amount of energy for the change of the path, for the change of the shape of the molecule. So in this case, it will have first need to go dephosphorylation, that means the removal of the inorganic phosphate group so that it can utilize the energy for this purpose and return back to its original state. And after which the K plus will bind and again the process will revert to its original conformation from E2 back to E1, where the K plus that were bind now will move to the inside, and again this process will keep on repeating itself. So here we can see in the overall reaction that three sodium ions which were present inside the cell and two K plus ions which are present outside the cell undergo hydrolysis along with the ATP molecule with magnesium at its active site. Then it undergoes dephosphorylation, utilizing the energy for transporting 3Na plus to the outside of the cell and importing 2K plus ions inside of the cell. Hence, we see that there is a net export of one ion from inside of the cell to the outside of the cell. So, in this case, since the charges are being transferred across the cell membrane, the concentration gradient will always be changing continuously, because of which the nerve impulses across our body continuously travel. That makes everything around our body makes us to feel everything around our body. So this was all about your sodium potassium pump. I hope this must be very clear and informative for you. And in case of any doubts, again, you have the comment section. You can add whatever doubts you have and we'll try to resolve them. Thank you.